All right, good morning, and I just want to thank the organizers and our collaborators at RTI for the opportunity to be here. Today, I'll be talking about the microbiome-metabolic interaction. So let's start with the human microbiome. So we are colonized on every surface, inside and out, with microbes, bacteria, viruses, and fungi. And there are 10 times more microbial cells than human cells. So we're actually much more microbe than human by number. And the microbial genes within the human microbiome, it's estimated that they contain 100 times more genes than the human genome. So there's a lot of metabolic potential that it's an environmentally acquired factor, and that can change our physiology. What's also important is that the microbiome is very niche specific. So the microbes on your hair and your skin are different than the microbes in your intestinal tract. And we've co-evolved to have stable microbiome communities. And they impart many beneficial functions such as pathogen displacement, immune system development, barrier fortification, vitamin synthesis, and short-chain fatty acid production. And so there's a little bit of a debate about this now, but it's generally believed that the fetus develops in a sterile environment and acquires most of the microbes at the time of birth from the mom. And that as the child matures, so does the microbiome. But there are forces of disruption, and so there can either be microbiome disruption on the maternal side, such as prenatal antibiotics, cesarean delivery, or antibiotics during nursing. And there can also be direct disruption of the infant and developing microbiome through direct antibiotic exposure. And early life microbiota disruption can alter immunologic development, behavior and neurobiology, and also growth and metabolism. So some of the earliest clues for this actually come from the agriculture industry. We've known since the late 1940s, if you give low-dose antibiotics to a variety of farm animals, it increases their feed efficiency and their growth rate. And you have more profound effects the earlier you give this. Importantly, this works across antibiotic classes, many different targets and modes of action. It does not work with antifungal agents. So we sought to develop a mouse model of microbe-induced obesity and to test the early life effect. So for this, we gave low-dose penicillin either at weaning or at birth by giving penicillin to mothers in their drinking water and followed them for 20 weeks. We examined their body composition by DEXA scanning and found that the mice that received low-dose penicillin from birth had significant increases in total mass, but we did not see that with the mice that received it at weaning. And in the male mice, we see significant increases in fat mass, and the effect is much more profound when they receive it from birth. Next, we looked at where the fat was going by MRI. And so, again, we saw elevated total body fat percentage. And when we look at the abdominal region, we find that it's significantly elevated visceral fat, not subcutaneous fat, more metabolically active. And we see significant elevations in liver adiposity. We also looked at hepatic gene expression and found significant elevations in 3 genes involved in lipid metabolism, PPAR gamma, CD36, and fatty acid binding protein. So we see systemic changes in liver gene expression. Next, we wanted to see if there was an interaction with high-fat diet before the mice were on a normal chow. And so this same experiment as before, we administered low-dose penicillin, and when they were 17 weeks of age, we switched half the group onto a high-fat diet. What we found is that we had significant elevations in total mass with antibiotics, but when we added the high-fat diet, as expected, both groups increased in total mass, but the ones that received microbiome disruption with penicillin gained even more weight. What was really striking is that female mice resisted weight gain on 45% fat diet. This is reported for C57 black mice. But when we disrupted their early life microbiome with penicillin, we saw significant increases in fat mass. And in fact, we saw a 100% increase in fat mass in the female mice. So all of the experiments I've shown you to this point use lifelong antibiotic exposure. And so we wanted to find out, is early life disruption enough? So in this experiment, mice received either 4 weeks, 8 weeks, or continuous penicillin exposure, and then we switched them onto a high-fat diet to accentuate the effect. And these results are in female mice. What we found, surprisingly, is that regardless of length of exposure, there was significant increases in total mass, increases in lean mass up until 20 weeks, and significant increases in fat mass after 20 weeks. And what this demonstrated is that early life microbiome disruption is sufficient for lasting changes in metabolism. We wanted to investigate which microbes were changing. So we sequenced the 16S rRNA gene, and we sequenced it on the MiSeq platform, and we used CHIME for our downstream analysis. And so what I'm first showing you here is a principal coordinates plot at 3 weeks of age. So each black dot is a control mouse, each orange dot is a mouse currently exposed to antibiotics. And it's projected in 3D space, and by ANOVA testing, there is significant clustering, so we can conclude penicillin changes the microbiome. That's not surprising. So when we jump to 8 weeks later, now we actually have 3 groups. Black is control, orange is exposed to penicillin, and the blue dots are the mice that were exposed to penicillin for 4 weeks, and then were allowed to recover for 4 weeks, taken off penicillin. And what's really interesting is that the mice that were taken off penicillin now cluster with controls. Their microbiome has recovered. And up in the top corner, I have a graph of the fat mass there. At 8 weeks, we do not detect changes in fat mass, but those mice go on to later develop obesity. And so this is our first evidence that early life changes in the microbiome can program host metabolism. And we had a second group that I took off antibiotics, and they too cluster with controls. Beyond just overall ecological changes, we wanted to find out who's changing. So we looked for microbes that were consistently altered at multiple time points and in multiple independent experiments. And what we found is that lactobacillus was significantly reduced, as well as Alobaculum, Reichenelaceae, and Candidatus arthromitis. Some of you might know it as segmented filamentous bacteria. And so we found consistent reductions in early life bacteria, and we think that these could be potentially new probiotic organisms to restore. We also wanted to know, is it the antibiotics or is it the microbes? And so we performed a microbiota transplant. I took cecal contents from control mice or cecal contents from mice receiving low-dose penicillin and transferred them by oral gavage to germ-free mice. They come in a bubble just like that. And what we found is that in just 5 weeks, the mice that received the low-dose penicillin-treated microbes gained significantly more total mass and significantly more fat mass. And this demonstrates that the altered microbiota is causing metabolic changes. We also looked by 16S sequencing to see if the same microbes were altered, and yes, we confirmed significant reductions in lactobacillus, Alobaculum, and Reichenelaceae throughout the experiment. So we wanted to find out what sort of metabolic functional changes were happening in our system, and so we paired up with RTI. We had a pilot and feasibility study, and so in this experiment we examined either mice directly exposed to penicillin or the germ-free animals that were either control microbiota recipients or low-dose penicillin microbiota recipients. And we looked locally where the microbes were, so we looked at the cecal contents, and then we also looked at the liver because of the enterohepatic circulation that can bring microbial and dietary metabolites to the liver and affect metabolism. We looked at the NMR spectra on the Bruker spectrophotometer, and then looked for significant peaks. So here is the PCA of the metabolomic profile from either mice receiving low-dose penicillin or the microbiota recipients in the liver and in the cecum. And in each of the PCA plots, you can see that there is some degree of clustering between control and penicillin. So the penicillin, the altered microbiota, is altering the metabolic profile in the liver and in the cecum. Now, in each of these different compartments, there were many, many metabolites that were altered, but instead I'll just focus on 4 that we found consistently altered throughout. So in the cecal compartment, we found significantly elevated ethanol. I promise you I was not giving alcohol to my mice, but we saw significantly elevated cecal ethanol, and ethanol can actually be a byproduct, a metabolic end product of certain microbes. And then in the liver, we saw reduced acetate levels. Acetate can have some effect on appetite. We saw significant reductions in glutathione and significant increases in hepatic maltose. In order to link this with some of the either microbial or host genes, we looked at the potential functional changes in our system using a tool called PyCrust. What this does is it constructs a predicted metagenomic profile from a 16S sequencing data set. And again, we were just looking for what are the consistent changes. And so what I'm listing here are the KEGG pathways that are either significantly downregulated or upregulated in 2 independent experiments at least 2 different time points within those experiments. And what we noticed, strikingly, is that there seemed to be a general downregulation of some carbohydrate pathways, glycolysis, fructose and mannose metabolism, and there was an upregulation in amino acid pathways and lipid pathways. And so this actually fit well with some of the metabolites that we detected. So if we have a decreased microbial metabolism of carbohydrates, that means that there could be more carbohydrates available for absorption, and that could potentially link with the elevated hepatic maltose. We saw other sugars as well. And then we also looked at liver gene expression by RNA sequencing, and we found in both mice receiving low-dose penicillin or low-dose penicillin-treated microbes, there was a significant upregulation in glutathione S transferase, and in the liver we see significant decreases in the substrate for that. And so to summarize, the key aspects of our model of microbe-induced obesity, in the host we see that early life is a critical window of host microbe metabolic interactions, and we see signs for increased need for liver detoxification or oxidative stress. In the microbes, we see consistent reduction in candidate protective taxa. We think that these microbes might be carrying out important functions. And we're able to find consistent metabolites between different experiments that also link up with other expression data. And finally, I'll just close on applications to the human population. So the model that we used was based on the agriculture doses, but there have been several independent studies that have examined antibiotics in early life and the risk of obesity later on. And many studies have found a significant risk if antibiotics are delivered within the first 6 months or the first year of life, and later increased risk for weight gain. With that, I'd like to especially acknowledge our collaborators at RTI and our funding sources and the many members of the Blazer Lab that participated in this work. Thank you for your attention.
That was a wonderful talk. Did you look at any behavioral cognitive changes in your mice?
No, we didn't, and I would really love to have done some of the behavioral changes. The only thing that we did measure was food intake by putting them in metabolic cages. We saw significantly increased food intake in early life, 6 to 8 weeks, but then later when the mice are already obese at 30 weeks, we see no change in food intake. We do see some changes in some of the metabolic hormones involved in appetite regulation.
And you probably didn't look at any potential epigenetic changes.
No, but there is a current postdoc, Chad Trent, in the Blazer Lab, and he has a whole project on that, so that should be coming out soon.
So if you affected the gram-negative microbiota, would you also see the same effects?
So actually what was interesting is some of our hits were gram negatives. Even though we were using penicillin, we saw a reduction in Reichenella-ACA, which is closely related to Bacteroides. And another colleague of mine tried different antibiotic classes and saw consistent effects regardless of antibiotic class. So we're not sure the particular role, but I would say that we're not lowering total bacteria, we're just having a shift. And so we're wiping out specific members.
And I guess the other question I have is, if you were to feed the antibiotics to the nursing animals, would that affect the weight gain of the animals?
Yeah, so in one experiment, we administered penicillin only during the time of nursing, and we gave it to the mothers, not the pups. And we were able to significantly shift their microbiota even though they were receiving the antibiotics through nursing.
Any other questions?
I've been playing with penicillin for a long time. I've worked with Dr. Chain, who was one of the Nobel laureates on penicillin. Have you ever put penicillin in a germ-free animal to see if penicillin is doing other things? Because penicillin gets absorbed and does not get metabolized. In the early days of its use, it was recovered from human urine and recycled and given back to people. So it's in the human system as is, and I just wondered whether it had any non-microbial effects. And anyone has ever thought about that?
Thank you. That's a great question. If penicillin has any effect on its own in germ-free animals. We have not done that in mice, but there was an experiment in chickens, and there was no change in weight gain in germ-free chickens, but there was a change in conventional chickens. And then Victoria Ruiz, another postdoc in the Blazer lab, recently gave Tylosin, therapeutic dose Tylosin, to germ-free mice, and there was no weight change. There was effects on immunity, again, different antibiotic, different pathways, but it seems that the weight effect is dependent on altering microbes.
I have actually two questions. One is, I'm almost sure you put the mice into an SPF facility? Mm-hmm. Okay. The second relates to the diet effects. You compared high fat with chow. Mm-hmm. Is this correct? Because the chow is undefined something. Mm-hmm. Yeah, and of course, when we talk about the role of microbiome to extract energy, we feed the mice a prudent, raw, starch diet. So they need the bugs to extract the energy. Mm-hmm. The story would be different in humans, except for people that just eat raw, prudent diets. Mm-hmm. So this is really chow, because then you have protein effects and soy polyphenols and all this kind of stuff.
Yeah, so for those, yes, unfortunately, it was chow. But for those, our direct comparisons, we're really examining control versus penicillin within the chow diet, and then control versus penicillin within the high-fat diet. Thank you.
Thank you.